This is Chapter 3, Part B on cells, the living units. So this section will look at different components that make up a cell and the process of cell division and protein synthesis. So the cytoplasm is all of the cellular material that's between the membrane and the nucleus in the core of the cell. So within the cytoplasm, you'll find cytosol, which is the main fluid component, kind of the goo in the cell. So it's made up of mostly water and then some soluble molecules and things like ions, proteins, salts, sugars. The organelles are the metabolic machinery of the cell. So they're basically like little organs that do different jobs and functions for the cell's overall health and metabolism, similar to our own organs. So there's several organelles within a cell that we'll look at. So they're divided into two basic categories. So you have your membranous organelles, which are a bit more complex just because that membrane allows for more compartmentalization. So a wider range of functions. And some non-membranous or more simple types of organelles and structures. The mitochondria is considered the powerhouse of the cell because this is where we derive our ATP energy. Remember, ATP is the primary energy molecule of the cell. It powers all of our cell functions. So ATP is generated from glucose in these little mitochondria. So these are membranous or membrane-bound organelles. So they have a double membrane that kind of folds in on itself on the inner membrane. And these folds are referred to as cristae. So this is just to increase the surface area of the mitochondria just so we can make more ATP with that limited space. So mitochondria are interesting because they're different from the other organelles that we're going to talk about because they have their own DNA, their own RNA, and their own ribosomes. So they also resemble bacteria. So kind of structurally, they look kind of like a bacillus bacteria, and they're also capable of the same type of cell division that bacteria can do. So mitochondria can replicate themselves, and they have their own separate DNA chromosome, and it's also a circular chromosome. So bacteria have circular chromosomes as well. Also, their ribosomes that make proteins are slightly different from our ribosomes. So our ribosomes also make proteins, but ours are a bit larger. They have the small ribosomes that are similar, again, to bacterial ribosomes. So this has led to the theory of endosymbiotic theory of eukaryotic evolution. And this also applies to chloroplasts. So chloroplasts in plant cells will also have their own DNA. They're able to divide via binary fission, and they have their own ribosomes. So this endosymbiotic theory of eukaryotic evolution states that there was a larger prokaryote or a larger bacterial cell that took up a smaller aerobic or oxygen metabolizing cell into its cell. So basically a large cell eats a smaller cell. But they have a kind of mutually beneficial or symbiotic relationship. Right? So endosymbiotic. So symbiotic just means mutually beneficial, and endo means inside. Right? So we have one cell living inside of another. So the aerobic bacteria would produce energy in the form of ATP for the larger cell, and the larger cell provided a safe place for the smaller bacteria to live. So over a long period of time, they just became dependent on one another and couldn't live separately anymore. So ultimately, this led to the development of our mitochondria, which now are just a permanent fixture in our cells and produce our energy, ATP, for us. And then this phenomenon happened again a second time with a photosynthetic bacteria. So same situation, the larger bacteria kind of engulfed and took in a smaller bacteria. And because they're photosynthetic, now they can produce sugar. So now this cell has its endosymbiotes to produce energy and sugar. So it's kind of a self-sustaining organism now. And again, over a long period of time, they just became kind of inseparable, and it has led to the evolution of chloroplasts that we see in plants today. Ribosomes are some of those non-membranous organelles, so meaning they're just more simple in structure, a bit more primitive, if you will, but they are very important. So ribosomes are the sites of protein synthesis. So every protein in your body is made by ribosomes. So ribosomes can be either free or membrane bound. So we'll talk about the membrane bound ones in a bit, but free ribosomes are free to just kind of float around throughout the cytoplasm and make proteins. The bound or membrane bound organelles are attached to another organelle that we'll look at in a minute called the rough endoplasmic reticulum. So the way ribosomes work is we have our mRNA, which is carrying the message of DNA, so kind of the instructions in the DNA. The ribosome will just kind of chug along this mRNA molecule and translate the message that's encoded in the genetic code, and it will assemble the appropriate amino acids to build our new protein chain. The endoplasmic reticulum consists of a series of parallel and interconnected and flattened membranous tubes continuous with the outer nuclear membrane. So there's two types of endoplasmic reticulum. We have a rough ER. So this one has those membrane-bound ribosomes attached to it. So this is why it's referred to as the rough ER, because it has this kind of rough textured appearance. The smooth ER has no ribosomes, so it's smooth. So because the rough ER does have the ribosomes, and we said the function of ribosomes was to synthesize proteins, a lot of proteins will be manufactured in this rough ER organelle. The smooth ER is going to focus more on synthesizing lipids, like for the plasma membrane, and also detoxifying other substances. So because the liver is our main detoxifying center of the body, the liver cells have high quantities of this smooth ER. The Golgi apparatus is kind of like the post office of the cell. So it's going to receive the proteins that were made by the rough ER and the ribosomes, and then it's going to modify, sort, and package these newly made proteins, and then ship them off to their final destination in a secretory vesicle. Lysosomes and peroxisomes are both similar in structure and function, but they contain different types of enzymes. So they're basically little enzyme sacs that the cell uses. Lysosomes are kind of like the stomach of the cell. So they're filled with these digestive enzymes that allow the cell to break down dead organelles or other cells. So when a cell eats or ingests another cell or a food particle via that endocytosis, right, so it'll kind of engulf this food particle, or say a bacteria, and form a food vacuole. So we kind of wall it off in its own little membrane sac inside the cell. So now our lysosomes that have been synthesized from the Golgi apparatus are going to bud off of here containing these digestive enzymes, and then they're going to fuse with this food vacuole. So the fusion of these two vesicles, the lysosome and the food vacuole, forms what's called a phagolysosome. This is basically the stomach acid now entering the stomach, so we can start the digestive process and break down these food particles. And also, just like in our own digestive system, anything that is undigestible will be expelled. So exocytosis and elimination of those waste products is essentially the same you could think of as the cell is pooping after it eats. Peroxisomes are similar, but they're going to focus more on detoxifying things like hydrogen peroxide or other harmful kind of waste products and free radicals that are produced just during normal cell metabolism. So this endomembrane system is going to consist of all of the membranous or membrane-bound organelles that we've discussed so far. So we have the endoplasmic reticulum, the rough and the smooth, then the Golgi apparatus, and our secretory vesicles like the lysosomes and the peroxisomes. It also includes the nuclear and the plasma membrane because they are also membrane-bound. So all of these membranes and organelles work together to produce, degrade, store, and export biological molecules, right? So things like enzymes would be produced by the ribosomes, since enzymes are proteins. Any waste products would be broken down by those lysosomes. We can also store nutrients and other things in vesicles within the cell. as well as export those. So we can store some proteins in the cell in a vesicle, and then when it's time to use those proteins, we can export them through this secretory vesicle. So this will just kind of bud off of the cell membrane. And because the vesicles are composed of phospholipids, just like the membrane, they can just kind of easily fuse together without disrupting the overall membrane structure. The cytoskeleton is an elaborate network of rods and protein filaments that run throughout the cell in this cytosol. So hundreds of different types of proteins will link these rods to other cell structures. So you can think of these kind of like the bones and ligaments and muscles of the actual cell body. So there's three main types of molecules associated with the cytoskeleton. The intermediate filaments are kind of like the basic framework and scaffolding of the cell. Your microtubules are kind of like the connection points, the bridges and the highways. So this is actually showing obviously computer generated model, but this is essentially what it looks like and how it happens. So this is a motor protein that is carrying one of those secretory vesicles, and it's walking along these microtubules to carry it to the other end of the cell where it needs to go to make sure it gets where it needs to go. Microfilaments, things like actin filaments, these are contractile proteins. They're also found in muscle cells. So these allow the cell to actually kind of move and contract different components. The centrosome and centrioles are a pair of microtubules that's going to be involved in cell division. So the centrosome is what it's called, or where it's located in the cell basically when it's not needed at that time, when we're not dividing the cell. So once cell division begins, we start the process of mitosis. Then some microtubules will expand from the centrioles and radiate out to kind of capture the chromosome. So the red is the chromosomes, the green are these microtubules in the centrioles. So during cell division, we have to kind of shift the chromosomes around to make sure everything is divided evenly. So these centrioles work to kind of capture the chromosomes in the cell so they're not just floating around, and that we can position them precisely like along the midline of the cell. So when we divide the cell, we have an equal separation of those chromosomes. Some cellular extensions include cilia, flagella, and microvilli. So cilia are like little small beating hairs on the outside of the cell. So these are primarily just to move fluid around the cell, not so much to move the cell through the fluid. So we have ciliated cells in our respiratory tract. So if you've ever had a cold and you get congested and you cough up a lot of that phlegm for a few days, that's the cilia in your respiratory tract lining that's kind of sweeping all of that mucus and debris out of your respiratory tract. The entire respiratory tract is coated with mucus, so any other dust or debris particles that you inhale will get trapped in that mucus and swept up and out. You'll either cough it up or it'll just be swept out into the stomach. So any bacteria that may have gotten captured in that mucus would be killed or expelled from the body and prevent those respiratory infections. So one reason people that smoke have that smoker's cough is because the cigarette smoke kind of paralyzes these cilia. So mucus and phlegm can build up more, and it's not as easily cleaned out. So this is also why when someone stops smoking, they usually have a worse cough right before it gets better. You would think if you quit smoking that your cough would get better, but it actually gets worse for the first week or so because the cilia will start to kind of come back alive and start cleaning out all of that accumulated tar and debris. Flagella are long whip-like tails that actually move a cell through a fluid. So our standard example is the sperm. So the tail on the sperm is called a flagella and allows the sperm to swim through the female reproductive tract to fertilize an egg. Microvilli are small cellular extensions that increase surface area for absorption. So these are found in the lining of the small intestines where we absorb our nutrients from our food. So if we look at the lining of the small intestine, you'll see it's not a smooth tube. It has all these ridges and folds in it. So this is another way to help increase our surface area. And then all of these ridges have these little finger-like projections on their surface, again, to increase that surface area. So if we were to take the small intestine and kind of flatten it out entirely, it would have the surface area of about the size of a tennis court. So it has very large surface area for maximum nutrient absorption. So then within these villi, if we look at the individual cells of the villi, the cells also have microvilli, again, just to increase our surface area for absorption. The nucleus is the largest organelle in the cell. So this one is usually easily seen under the microscope. So this is a slide of a cheek cell. So just a simple squamous single cell, but the nucleus is clearly defined kind of in the central core of that cell. The nucleus contains the genetic blueprint of all life. So basically all of the instructions and the recipe of how to make one of you is in your DNA in the nucleus. Most cells only have one nucleus, or they are uninucleate. So like this one here only has one nucleus. But some cells, like skeletal muscle, and some bone cells and liver cells, may be multinucleate, meaning they have many nuclei within one cell. So we'll look at muscle cells later in the semester, but muscle cells are kind of very long, thin fibers, fiber type cells. So they have multiple nuclei within one cell just because they have a high demand for proteins. There are also some cells that have no nucleus. So red blood cells are actually anucleate, meaning they do not have a nucleus. So if we look at blood smear under the microscope, sometimes they look kind of like little red donuts. So this white in the center of the blood cell is where the nucleus has degraded, and it's just kind of an empty open space now. You can also see, though, that the white blood cells, so these two big ones here, these two big purple ones, they do still have their nuclei. So this is a neutrophil. They have kind of an oddly shaped nucleus. It's not a perfect sphere like in the epithelial cell. So the nucleus has three main structures. The nuclear envelope, which is going to serve as kind of that membrane, that barrier, selective barrier similar to our own cell membrane. So it only allows certain things in and out to have access to the precious genetic material inside. The nucleolus is the site of ribosome assembly, ribosome synthesis. So we said ribosomes are the site of protein synthesis. Well, the nucleolus is the site of ribosome synthesis. And chromatin is just kind of the loosely bound DNA that's within the nucleus. So it's just kind of loosely bound in there to a few proteins, kind of like spaghetti noodles. So this is okay when, right, we're wanting to synthesize protein. So we want to be able to access that particular gene on the DNA and the chromosome, right, to make that protein. So normally when you see pictures of chromosomes or DNA in a cell, right, it's in these little X-shaped chromosomes. Normally, if the cell is not dividing, it's just kind of doing its own everyday cell thing, the DNA will be in this loose chromatin arrangement. It's only before the cell prepares to divide and make a copy of itself that it packages the DNA into these tight little X-shaped chromosomes, just to make sure we don't leave anything behind. So chromosomes are just condensed chromatin. So again, this condensed state just helps to protect the threads during cell division. So we just package it nice and tight so we don't leave any genes or any sections of DNA behind in the cell. So the way that we package about 6 feet of DNA into these tiny microscopic chromosomes is through coiling around proteins. So here we have our kind of double helix DNA strand. So then it's going to wrap around these proteins called histones. Right, so we'll have a few of these bundles of DNA and proteins, and then they'll start to coil around themselves, right? Kind of package even more densely. And then the coils will start to coil and form the supercoils, eventually until we get that tight X-shaped chromosome. So here's the histones wrapping the DNA, and then here's the nucleosomes starting to coil, and then we have our finished final chromosome. The cell cycle is a series of changes a cell undergoes from the time it's formed until it reproduces. So basically, just like us, cells have their own life cycle, their life stages, right? So how we have childhood, adolescence, and then adulthood, right? Cells have their different life stages or phases. The two major periods of the cell cycle include interphase, which is basically when the cell is just carrying out its normal everyday cell life. It's not reproducing, it's growing, right? It's getting its affairs in order, right? It has to finish school first and get a job before it's ready to reproduce. So the cell division phase, or the mitotic phase, is only a small fraction of the cell's overall lifespan. Again, similar to humans. Right? So we don't spend the majority of our life reproducing. Only a small fraction of our life would be spent, right, being pregnant and reproducing. So during the mitotic phase, we're going to divide the cell in two and make copies of the DNA. So the end result would be two brand new identical cells. So we basically clone our original cell and make copies of it. So DNA replication occurs during that interphase portion of the cell cycle. So it results in two new identical daughter strands that are formed using the original DNA molecule. Because of the mechanism of replication, each cell will have half of the original DNA and half of a new DNA. So this is called semi-conservative replication. Right? So if something's conservative, you kind of want to keep what we have, right? What we already have, conserve what we have. Semi just means kind of. So we're kind of keeping the old while bringing in the new. So we have our original molecule. And again, when we talked about the structure of DNA, this double helix structure is due to this replication mechanism. Unwind the DNA. So now we have our two original red strands. So these bases are exposed and can bind to form a new strand because remember the complementary base pairing rule. So A always binds with T, C always binds with G. So if we only have one half of the molecule, we can still figure out what the second strand of the DNA molecule would be. Right? So we have C, so we add a G, right, to form our new strand. So we'll do this for both copies. Right? So it's semi-conservative. So we get one old strand in our new molecule and one new strand. Most cells need to replicate continuously for growth and repair purposes. So think of the skin on the inside of your mouth. It's constantly undergoing abrasions or getting burned. Right? So we're constantly replacing those cheek cells on the inside of your mouth because they're constantly getting damaged and sloughing off. There are some tissues, though, and some cells that cannot divide efficiently. So things like cardiac muscle and nerve tissue. So these cells, if they become damaged, generally would be replaced with scar tissue instead of undergoing mitosis and replacing the original cells. So think if someone has a heart attack, so if that cardiac muscle dies, it's not going to grow back. So it's going to be replaced with scar tissue, and scar tissue is not muscle tissue. So muscle tissue is able to contract, right, and help pump the blood. Scar tissue can't contract. So after a heart attack event, the damage is permanent and irreversible. So the heart will never be 100% fully restored to its original function. So this mitotic phase or cell division phase has two distinct events, even though the majority of it is in the mitosis. The cytokinesis is just kind of the very last step. So mitosis is the division of the nucleus. So we're physically separating the chromosomes and the nucleus. So there's 4 phases of mitosis that we'll look at in order of PMAT. And then cytokinesis is this last phase. So this is division of the cytoplasm. So once we've separated the chromosomes and the nucleus, then we kind of pinch off in the middle to separate the two cell membranes. So the control of the cell division process is crucial. So cells only need to divide when necessary. We don't want them to divide unnecessarily. So that could lead to things like cancer growth. So an uncontrolled cell growth is a type of cancer. So looking at the phases of mitosis, so we said PMAT was the order. So prophase is the first step of mitosis. Remember, the DNA is in the nucleus, but we're trying to separate the DNA, right? We want to divvy up the DNA into the two new daughter cells we're about to produce. So first step is we have to disintegrate and break down this nuclear membrane. So we break down the nucleus to free up the chromosomes. So now they're kind of floating around in the cell. And also the mitotic spindle from those centrioles and the centrosome will start to form. So these microtubules will expand outward to catch these chromosomes so they don't like float away. So we can position them precisely where they need to be during this process. No chromosome left behind. So second step is metaphase. So M for metaphase, this is when the chromosomes line up in the middle, right? So M for middle. So metaphase, these spindle fibers, they have captured the chromosomes and now they're going to position them along the midline of the cell. So when the cell starts to divide in our next phase, we want an equal division on both sides. We don't want a lopsided division, a lopsided or unequal number of chromosomes. Step 3 is anaphase. Right? So anaphase is when the spindle fibers are now going to retract and separate the chromosomes, right? Pulling them to opposite ends of the cells. Right? So remember A for anaphase, A for apart. So this is when they're pulled apart. So metaphase, M, they're in the middle. Anaphase, now they're being pulled apart. So last step is telophase. So during telophase, the chromosomes have moved to opposite poles or ends of the cell. Cleavage furrow will begin to form, so it just starts to pinch off in the middle of the cell. And also the nuclear membrane will start to reform. So now we've separated the chromosomes, we need to repackage them back in that nucleus so they'll be protected. And then finally, cytokinesis is that very last step where we separate the cytoplasm. So we've reformed the nucleus. Again, just to review the 4 phases of mitosis, right? PMAT. So you can come up with your own little mnemonic or sentence to help you remember these in order, whatever works for you. You say something like Prince made awesome tunes. So to review the phases of mitosis, so prophase, metaphase, anaphase, telophase, PMAT. So prophase, the nuclear membrane breaks down and the spindle fibers form. Metaphase, we line the chromosomes up along the midline of the cell. Anaphase, they're going to be pulled apart. And then telophase, we reform the nuclear membrane and start that cleavage furrow, start to separate right along the middle. So this figure is an actual cell undergoing mitosis. So it's been injected with some dye just so we can see it. So we have metaphase, anaphase, telophase. So it happens really quickly, but you can also see the formation of the spindle fibers and the movement of the chromosomes. And then right as soon as this one finishes, now the neighboring cell is going to begin. You can kind of watch it and see right before it kind of implodes, you can see that nuclear membrane start to break down and it gets kind of a grainy appearance. The checkpoints are going to be the key events during this cell cycle to ensure that everything is running smoothly. So we don't want any kind of unmitigated cell division. So there's different checkpoints along the way. But G1 is probably going to be the most important of the 3 checkpoints. So this is our first checkpoint before we synthesize the DNA. So the S phase is for DNA synthesis. So we want to make sure our DNA is intact, right? And we have all of our chromosomes in order, everything's packaged correctly. We don't want to leave any DNA material left behind during this copying or synthesis process. So normally the cells are pretty good at catching these kind of errors, but occasionally mistakes do get through, and that's when you have things that can lead to cancerous growths, uncontrolled cell division. So after the S phase synthesis, we go into G2, which is just the second growth phase. So we're kind of getting our ducks in a row before we get ready to divide. So we have one more checkpoint to make sure we've completed our DNA replication entirely. We didn't leave anything behind. We don't have any mutations in our copying process. We didn't make any mistakes. So then the third checkpoint is during the mitotic phase. So in between metaphase and anaphase, before we separate those chromosomes to the opposite ends of the cell, we want to make sure that they're all aligned up perfectly in the middle. So if we have any kind of off-kilter alignment, that could lead to an unequal number of chromosomes in the daughter cells. So protein synthesis is kind of the central dogma of all biology. So how do we go from the genetic code in the DNA and the nucleus of a cell to an actual functioning three-dimensional organism and proteins? So we know DNA is kind of the genetic blueprint that holds the instructions for all of our protein synthesis. So all of the recipes for all of your proteins are in your DNA. So the segment of DNA or a specific region of DNA that codes for one complete protein is referred to as a gene. So you can have thousands of genes on the same chromosome. Right? They'll have defined start and stop regions. So you could have a gene for hair color, there's a gene for insulin production, a gene for hemoglobin. So actually, sickle cell anemia is the result of a mutated version of the hemoglobin gene. So it's basically just one letter in the gene sequence has been changed, which changes the overall structure of the hemoglobin protein and the red blood cells and their function. So everything ties back into that structure and function complementarity. So the genetic code is determined by the specific order of those bases, the ATCG, in the DNA. So genetic code is read as 3 bases at a time. So these are called codons. For example, GGC codon, so those 3 bases in that order would code for the amino acid proline. So if we just change one of these letters, so now it's GCC, that's going to code for completely different amino acids and lead to a completely different protein. So each codon is specific for a particular amino acid. So we said that there were 20 amino acids. So we're not going to get too in-depth into this in this class, but there are multiple codons for the same amino acid. So this is called the degeneracy of the genetic code. It's kind of a built-in safeguard against some of these minor mutations. So there are some examples where if we just changed one letter of the codon, it would still code for the same amino acid, so it wouldn't change the overall function and structure of our protein. But you can think of it kind of like in terms of a language. So the language of DNA. So similar to our own language, our alphabet has 26 letters. So we can arrange these letters in different orders, right, to make different words. So it's the sequence or the order of the letters. So with DNA, we have 4 letters in our alphabet, those 4 nucleotides, A, T, C, and G. But with these 4 nucleotides, we can make 20 amino acids. And with these 20 amino acids, we can make over 100,000 proteins. So just like our words represent certain images, these DNA words or codons represent certain amino acids. So CAT to us would just mean cat. GGC to DNA means proline. We talked about RNA before in that it is a nucleic acid similar to DNA, but with some important differences. RNA is kind of the temporary copy of the genetic information. So it's going to carry that message, that information, to the ribosome. So that way we can translate that into protein. So we said one difference between RNA and DNA is that RNA is single-stranded, right? Well, DNA is double-stranded because we need that mechanism of replication. So that semi-conservative replication, right, is due to this double helix, this double-stranded structure. Since RNA is just a temporary copy, like a temporary message, we're not going to be replicating this DNA, so we don't need another strand. So once we've translated this message and made our protein, then we're done with this RNA. Another difference between RNA and DNA is RNA uses uracil in the place of thymine. It's going to function exactly the same in that it will pair with adenine, but that's just one way we denote the difference between DNA and RNA is the uracil replacing the thymine. RNA has ribose sugar instead of deoxyribose. So we said all nucleic acids are formed from nucleotides. Right? A nucleotide is a sugar, a phosphate, and a base. So the sugar in DNA is a deoxyribose. The sugar in RNA is ribose. So we get the D in DNA and the R in RNA. There's 3 types of RNA. So messenger RNA is the primary type of RNA. So the single-stranded messenger. So this is carrying essentially a copy of the genetic code in the DNA that we can use to translate into protein. Ribosomal RNA is what's going to physically make up the structure of the ribosome. So the ribosomes are what is going to read this message and translate it for us. The transfer RNA or tRNA are going to transfer our amino acids to our growing protein chain. So transfer RNAs or tRNAs have anticodons, right? So we said the genetic code is read 3 letters at a time. It's called a codon. So the way that we know which amino acid is coded for by each codon is because the tRNA will have an anticodon that will complementary base pair to our mRNA and deliver that amino acid. Protein synthesis occurs in 2 steps. Transcription occurs in the nucleus. So it's where we make a copy of our gene into mRNA. So DNA information is encoded into mRNA. So DNA is in the nucleus. DNA cannot leave the nucleus. So this is another reason why we need RNA. So it's kind of our middleman, our intermediary form of that information transfer. So RNA can leave the nucleus. So we'll make our copy of the gene, we'll get our information, and then it exits through the membrane, through a nuclear pore, where then it will find a ribosome. So step 2 is translation. So we have our message, now we need to translate. Well, what protein is this coding for? So again, the mRNA is just carrying the message, the genetic code. The tRNA is what's delivering our amino acids. These anticodons will bind to our codons, and we'll add those amino acids to our growing amino acid chain. So I always like to use the analogy of making a cake. I like to bake. So you think of protein synthesis as you want to make a cake, right? But you don't know how to make a cake. You need a recipe for a specific type of cake. So say we only want to make a chocolate cake. So we'll go to the library, right, where there's lots of books, lots of cookbooks, right, lots of information about how to make cakes. So we go to the library and we find our cookbook, our DNA in the library. But this library is particular, so you can't actually check out any books from this library, but you can make photocopies of particular pages, right? So we only want one recipe for chocolate cake. We don't want to make every type of cake in our cookbook. So we'll just make a photocopy of that one page, that one recipe that we're interested in for chocolate cake. So we can take our copy home with us out of the library or the nucleus. So then we go back home to our kitchen in the cytoplasm, and we get all of our ingredients together. So our amino acids are the building blocks of protein. So this would be our ingredients. And the ribosomes are what synthesize and assemble those proteins. So this is going to be our mixing bowl. So we mix all of our amino acids together by following the recipe, right? So we put them in the correct order, step by step. And our finished product that we have is a cake, right? Or our protein. So we have, how did we go from, words on a page, right, or just instructions, to a 3-dimensional functional cake that we can actually eat and enjoy.